Hi, my name is Bryan, and I'm a PhD student here at Arizona State University, and I am a part of Press A Lab. In Press A Lab, we use the latest tools from Bayesian machine learning to solve complex problems within biophysics. Today, I'm going to be telling you about one of these tools that was recently accepted for publication by Nature Computational Science. I'm excited to tell you about it, so let's get started. This particular project deals with fluorescence microscopy. Now, fluorescence microscopy is a great way to generate contrast within a cell. However, when you have two fluorescent labels, or fluorophores, that are within close proximity to one another, the diffraction limit of light makes the spots that you see from either fluorophore overlap, so you cannot distinguish between two fluorophores under a microscope. Additionally, when you see a spot, the brightness of the spot correlates to the number of fluorophores within the spot. However, noise artifacts such as photon shot noise and detector noise make it difficult to distinguish the number of fluorophores within a spot based off of brightness alone. Now, there are a class of experiments called counting by photobleaching, which hope to overcome these challenges by illuminating a spot with constant laser power and looking for step-like brightness drops as the fluorophores photobleach one by one. This is a great way to overcome some of these challenges. However, this still presents several sources of difficulty. In particular, photophysics, like blinking, can make steps appear as the fluorophores blink and unblink, such that the number of steps you see in the experiment does not correlate to the number of fluorophores. Additionally, the noise scales with the number of fluorophores, meaning that while spots can be resolved very easily when you have a few fluorophores, like 3, when you have 100 fluorophores, suddenly steps are much harder to see by eye, and often become impossible to see by eye. Lastly, fluorophores might start dark, so the initial brightness you see at the beginning of the trace does not indicate the number of fluorophores within the ROI. Now, this is a picture of a simplified photobleaching experiment, but when you have large numbers of fluorophores, you might see something like this, where now these steps are no longer visible by eye, and we need to use other methods to infer how many fluorophores are within this ROI. So that is the problem that we are trying to tackle today, and now I am going to explain one approach that our lab has created to solve this problem. Now, because the processes that create the data are largely stochastic, we rely on Bayesian tools to infer the number of fluorophores that are in each ROI. And because the number of fluorophores could in principle be infinite, we rely on a class of tools called Bayesian Nonparametrics to solve this problem. The Bayesian nonparametric formulation is outlined like this. We start with a detailed generative model that explains how the parameters, and particularly the number of fluorophores, the brightness of the fluorophores, the transition kinetics, they all give rise to the data that we see. Then we use Bayes' theorem to create an inverse model, which allows us to sample the parameters that we care about from the posterior of the parameters given the data. Once we have this sampling scheme, we generate lots and lots of samples using efficient tools like Gibbs sampling and Hamiltonian Monte Carlo. And then lastly, we can use our samples to generate estimates of the desired quantities. So let's go into these each in greater detail. Starting with the forward model, we model the dynamics of a fluorophore using a hidden Markov model. So basically we assume that at each time level a fluorophore can either be dark, active, which means it's bright, or photobleached, which means that it has irreversibly gone into a dark state and will no longer fluoresce. The state of the fluorophore at each time level is conditioned on the state of the fluorophore at its previous time level, according to a transition matrix that we also wish to learn. The brightness that we measure is conditioned on the number of fluorophores at the time level that are in a bright state. Here we build a graphical representation of the model. Starting with a single fluorophore, we note that we have one random variable for the state of the fluorophore at each time level, 1 through n. These states are linked by a transition rate matrix which governs the transition probabilities between time levels. At each time level for a single fluorophore, we are given a measurement which is conditioned on the state of the fluorophore at that time level. These measurements are also conditioned on the brightness of the fluorophore in the background. Because we have multiple fluorophores, we need to denote this by adding a plate behind the states of the fluorophore, which indicates that we have one fluorophore for each K in the index. Lastly, we analyze multiple ROIs at the same time, so we add an additional plate around all variables that vary per ROI. So now that we have a forward model, to create an inverse model, we put priors on the variables that we wish to infer. In particular, we put priors on the transition matrix, we put priors on the fluorophore brightness and the background brightness. We cannot put priors directly on the number of fluorophores K, as K is an index that contains all of the fluorophores within its plate. So in order to sample the number of fluorophores, we rely on a trick by invoking what's called a beta-Bernoulli prior. The beta-Bernoulli prior allows us to sample the number of fluorophores as follows. So here we have the forward model that I have told you before. What we want to do to sample the number of fluorophores is we're going to start with a number of fluorophores that is much, much greater than the number of fluorophores that we expect to encounter. So K in this particular case is going to be much larger than the K we expect to encounter. But we allow some of the fluorophores to not exist and some of the fluorophores to exist. In the existence of a fluorophore, we encode with a variable called a load, B. B can either be on or off. If B is off, then that means that this particular fluorophore does not contribute to the brightness that we measure. If it is on, then it does contribute to the brightness that we measure. And we have the expectation that by sampling the loads, B, we will flicker fluorophores in and out of existence as warranted by the data. So these loads allow us to sample the number of fluorophores in an ROI by flickering on and off as warranted by the data. By summing the number of loads that we have, we in turn get the number of fluorophores in an ROI. Now I'm going to show the results of our method. So our collaborators at Dirk Peter Hurton Lab in University of Birmingham provided us with data from DNA origami experiments. We were given two data sets, one with DNA origami containing 20 binding sites and one with DNA origami containing 35 binding sites. There was a 70% labeling efficiency, so we expect a binomial distribution in the ground truth number of fluorophores, meaning that we can compare our inferences against a well-controlled ground truth. As you can see here, the estimates provided by our method match neatly with the estimates from the binomial distribution. The mean almost exactly overlaps with the expected mean. We have a larger uncertainty, but that is largely due to the fact that our estimates are going to have uncertainty in each, so the full posterior will be wider than the actual binomial distribution, just because it's going to be a convolution of the ground truth number of fluorophores with the uncertainty that we have from our method. Now, we have 35 and 20 binding site data, but we can add up to the brightness of 2 ROIs in order to simulate a semi-empirical data set with a larger number of fluorophores. For example, by adding 2 20 binding site data sets, we can create one 40 binding site data set. So, analyzing the semi-empirical data, we can show that our method allows us to count up to 100 fluorophores very accurately. We can also verify our method on simulated data to allow conditions that were not provided in the experiment. For example, we simulate data in which some of the fluorophores start dark. Now you can see on the right here, we compare our method to many other existing methods for treating counting by photobleaching experiments. These other methods do not take into account that some of the fluorophores can start bright, whereas ours does. For that reason, our method is able to infer a more accurate number of fluorophores than the other existing methods. Similarly, we can create a data set that has higher noise. Now, higher noise in a real setting could be achieved by lowering the laser intensity and increasing the gain to increase the photon shot noise and detector noise. Here again, we're able to count the fluorophores rather accurately compared to other methods. So the advantage of our method is that we can count upwards of 100 fluorophores. In addition to that, because ours is created using a Bayesian framework, we propagate uncertainty, and so we can get uncertainty bounds on all of our estimates for the number of fluorophores as well as the rates and the brightness values. Not only that, but we can infer the trajectories of the fluorophores, which could be extrapolated in the future to infer kinetic rates from binding and unbinding. If you were to take blinking and unblinking as a proxy for binding and unbinding, this would potentially be a method that could learn binding kinetics. Additionally, our method learns the photophysics and the photophysical transition rates on the fly, which eliminates the need for pre-calibrating dyes with other experiments. Lastly, because the fluorophores do not need to start bright in our method, this eliminates the need to finely tune the photophysics of the fluorophores, which means that this opens up opportunities for more experiments in less controlled environments, for example, in vivo experiments. Thank you all for listening. If you enjoyed this talk, be sure to check out our paper appearing in the February edition of Nature Computational Science. I'd like to thank our collaborators, Dirk Peter Herden, as well as our friends at the NIH and the NSF. Thanks for watching. Have a great day.